The microbial genetics of gene expression. And it starts with the main players, the structure of a gene promoter. And so the first thing you see here is a big RNA polymerase, which will use DNA as a template to make an RNA copy. In this particular case, since we're talking about gene expression, a messenger RNA copy. Here's a hypothetical promoter with a couple important features pointed out. The first is the numbering system. So whenever we're looking at upstream, which means in the opposite direction of transcription, everything is numbered in negative numbers. So +1 then would be the very first A in the start codon, ATG. That would signify where transcription initiates. The downstream, anything downstream of that is all positive numbers in terms of numbering, so that's why the first A is a +1. But then if we go upstream, everything is numbered as negative numbers. So the -10 region, also called the Tata box for obvious reasons, because that in different organisms, the -10 region always seems to have a mixture of A's and T's that spell out Tata, at least somewhere. And then if you keep going upstream, you find the other region, the -35 region, which roughly has this consensus sequence between different microbes. So different points of contact for the RNA polymerase occur in the -35 and the -10 regions. And then obviously there are additional features, but I'll talk about those in subsequent slides. So this enzyme, the RNA polymerase, is a multi-subunit protein consisting of a core enzyme, which is 2 alpha subunits, a beta and a beta prime subunit. An additional protein subunit called sigma attaches, and when all 5 of those, and there's more subunits, but when these are the main ones, when these main 5 subunits come together, they form the holoenzyme. This is the enzyme that can actually undergo transcription. This is just a little bit about regulation of transcription initiation. And so some genes are always turned on. They are usually housekeeping genes, and so they might encode ribosomal RNAs, for example, that would be used to make ribosomes. These are called constitutive genes because they're always needed, and so they're always on. Inducible genes, however, are genes that are only needed under certain circumstances, so they can be induced on or induced or repressed if they're not needed. And so these are examples of that would be genes that encode inducible enzymes like beta-galactosidase. Beta-galactosidase is necessary for the utilization of lactose. So not surprising, if the microbe doesn't have any lactose around to eat, then it would turn off a gene like that. Whereas if there suddenly is lactose in the environment, the microbe might want to turn that gene on. So a little bit about beta-galactosidase. It's an inducible enzyme and it functions in the catabolism of lactose. The reaction is catalyzed by lactose hydrolysis into galactose and glucose. That's the reaction that it performs. So lactose, the disaccharide, is hydrolyzed into galactose and glucose. The level of enzyme can rise significantly, from about 3 molecules to maybe 3,000 molecules in the presence of an inducer. So the inducer in this particular case is some breakdown product of this reaction. And in this particular case, it's an isomer that I'll show you a picture of. So another thing that you need in order to eat lactose is a lactose permease. And so not surprising, this permease is also an inducible enzyme. And so the lactose permease, when lactose is present, will facilitate the uptake of the lactose into the cytoplasm using a proton to uptake the lactose. So when the disaccharide lactose comes in, the beta-galactosidase enzyme will split it here at this glycosidic linkage, and there's your galactose and glucose, and then the microbe can ferment these. There's also a side reaction that beta-galactosidase produces, and it forms this isomer called allolactose. And allolactose is actually the inducer that I alluded to previously. So when the levels of this build up, then the lactose permease and beta-galactosidase activity increase significantly. And I'll put that all together for you soon. There's negative control is important as well, and obviously there's regulatory features of the DNA to facilitate this. So there's repressor proteins that look specifically for operator sites on the messenger RNA, and these operator sites are where negative regulation begins. So a repressor would sit on an operator to shut down gene expression of a gene that doesn't need to be turned on at a particular time. And so repressors can actually exist in multiple forms, active and inactive, and inducers and co-repressors alter their activity. There's also a positive control component. Usually the positive and negative control components are competing with each other in order to get fine-tuned gene expression. So the presence of a regulatory protein or activator protein at a regulatory region promotes transcription. So there are positive regulators that activate lactose operon. These are CAP, or catabolate activator protein, and another component called cyclic AMP. So these two together, this cyclic AMP binding protein called CAP and cyclic AMP together will form an activator. So so the cyclic AMP receptor protein, or CRP, and cyclic AMP together will form the positive control. So it looks a little bit like this. This panel shows negative control of an inducible gene. So an inducible gene is one typically that's involved in catabolism, the breakdown of some substrate. And so if the substrate's not present, then this would be the default setting. You'd have an active form of the repressor that would sit on an operator and shut this down so you don't get transcription. However, if an inducer is present, then the repressor protein is knocked out. That allows transcription to occur. So in this particular case, the inducer could be that allolactose that I showed you in the previous slide. Here's negative control of a repressible gene. So usually these guys are, these repressible genes are typically like biosynthetic operons, something that they use, something that's needed to make some important component of the cell, not break something down that you take in from the outside world. So the repressor protein is synthesized inactive. It's called an aporepressor. It's not active. And so the default setting is for transcription to happen. But eventually, you can get some kind of a co-repressor to bind to the repressor to shut things down, and so you get no transcription. So a co-repressor could be an end product in that biosynthetic pathway. If you've got enough of that end product, you can then shut down the biosynthetic pathway. So competing with those negative reactions are positive control reactions. So in this particular case, there's an activator protein, and the activator protein isn't active unless it needs to be. And so again, the default setting in those inducible type genes is for no transcription, because again, usually they're catabolic, and if the catabolite is not present, then there's no need for turning those genes on and wasting energy. But again, if some inducer is present that can activate the activator, then this can sit down on the promoter and allow RNA polymerase access to the promoter and initiate transcription. So more specifically, allow RNA polymerase to see the -35 and -10 regions that I talked about, and then facilitate transcription. Here's positive control of a repressible gene. And so, and you can put these kind of together, this side by side with this, for example. So you can see these competing processes. The activator protein in this particular case is active and sits right on its little binding site on the promoter, allowing RNA polymerase to do its job. And then, of course, there's also the possibility of an inhibitor knocking out that positive regulator. The concept of operons is really important in microbiology specifically, because microbes will often use operons, which are multiple genes that are regulated by a common promoter. And this is a way for microbes to rapidly increase gene expression. Rather than having to turn on multiple promoters linked to individual genes, they can turn on a single promoter linked to multiple genes that are necessary to work together. And so the lactose operon, or lac operon, is an example of that. Here's just a kind of a brief conceptual organization of an operon. So again, 2 or more genes regulated by a single promoter. So here's our promoter, and then here's multiple genes. And imagine these genes A, B, and C conspire to have a common function. And so, again, this could be for the utilization of lactose. So imagine here's the promoter, and then genes A, B, and C all help to break down and utilize lactose. And so then you would want those genes to be co-regulated. So the messenger RNA that would result would have a ribosome binding site, followed by an open reading frame that the ribosome can read to make an amino acid sequence or protein, and then followed by another ribosome binding site, and then another open reading frame that can create the next protein, and so on. Sometimes these operons are 10, 12 genes long, if it's a more sophisticated process like biofilm formation. So this slide then gives you, kind of ties together a lot of those concepts. Regulation of the lac operon by the lac repressor. And so some of the features of the operon, there's the lac repressor gene, lac I. Here's the lac promoter, lac P. Lac operator, lac O, where the repressor would bind. And then Z, Y, and A genes of the lactose operon needed for the breakdown of lactose and uptake. So lac Z is the beta-galactosidase gene. Lac Y is a permease. And then lac A is a transacetylase, a gene that probably helps in the uptake of lactose. So this is the permease, and this is the beta-galactosidase, once again. And so again, the repressor is synthesized in this particular case. The lac repressor is synthesized active, right? Because there's no lactose in this hypothetical scenario, and so the lac operon is shut down. The lactose repressor finds the operator and turns this off, because there's no lactose in the environment. However, if there is lactose in the environment, if there is lactose in the environment, oh shoot, there we go. Lactose is present in the environment, then we'll get a buildup of allolactose. Remember that little isomer that beta-galactosidase will make, which will bind to specific regions of the lactose repressor, the lac I repressor, and it won't be able to find the operator. That allows the RNA polymerase to sit on the promoter and transcribe the beta-galactosidase, lactose permease, and transacetylase, which I just mentioned. And so that allows for utilization of lactose. So this is just a little bit of the animation of the operon. And so again, we see the genes for lactose utilization. Here's the promoter. Here's the genes for lactose utilization, Z, Y, and A. And so the negative regulator site is the operator here, and the positive regulator site is the cap binding site. And so there's cap, the lactose promoter, and then the operator. And so here's so if glucose levels are low, and that means that there's a high amount of, that means that there's a high amount of cyclic AMP, then cyclic AMP will bind to the cap protein, and those two will sit on the cap site, and the RNA polymerase will be able to bind to the lac operon, or the lac promoter here. If glucose levels are high, however, and that means that there's low cyclic AMP. Now, I should have backed up and said that the microbe is going to prefer to eat glucose. So if you give the microbe glucose and lactose in the same test tube, and then add E. coli, for example, the microbe will eat up all the glucose first and then eat the lactose later. It's just like a kid. If you put ice cream and broccoli in front of them and tell them to eat whatever they want in whatever order, as long as they eat it all, they're going to obviously eat the ice cream first. But so microbes are able to do that, and one of the ways they do it is through this process of sensing cyclic AMP. But in this particular case, glucose levels are high, so they're going to want to eat glucose first and shut down the lac operon. And so in this particular case, the cap protein doesn't have cyclic AMP to bind to, and so cap doesn't get bound. The RNA polymerase can't recognize the promoter as easily. And so again, if glucose levels are low, but lactose is absent, then the repressor protein sits on the operator and shuts things down. Remember that in order for this to not happen, you need the allolactose from that lactose reaction to knock out this guy here to stop it from binding. So in this particular case, if glucose levels are low but lactose is present, then cyclic AMP and CAP can do their job. The lactose repressor gets knocked out by the lactose isomer that I showed you before, and then the polymerase can sit on its site and then transcribe the lactose operon utilization genes. So a lot of gene expression relies on two-component regulatory systems and phosphorylated systems in bacteria. And so I'll talk a little bit about those. The prototypical example of that would be the regulation of porin proteins by two-component signal transduction systems in bacteria. And so porin proteins are these outer membrane pores that occur in gram-negative bacteria, and they allow dissolved solutes from the environment to come into the cell. If you let too many of those dissolved solutes in, you'll have an osmotic imbalance and the cell can burst or shrivel up. And so it's important to have the right proportion of outer membrane porins. The OMP-F porins that you see here are favored in dilute environments because they have very large pores. So they allow more solutes to enter. So if you're in a dilute environment, you want big pores to allow the very small bits of solutes to come into the cell so you can balance across the membrane. If you're in a high osmolarity environment with lots of dissolved stuff, then you want OMP-C, smaller porins. And so the microbe can actually regulate the proportion of these depending on the conditions. And so in this hypothetical example, there's a high osmolarity environment, so lots of salt or lots of sugar or something in the environment. And so obviously the microbe would want these more OMP-C than OMP-F. And so in this particular case, solutes come in, and that high solute concentration here in the periplasm activates this two-component system that you see here. The first component is MZ, which is called a sensor kinase, and the second component is OMP-R, which is called a response regulator. So excess solutes here causes MZ to phosphorylate at this key histidine amino acid here. So ATP will give up a phosphate group right here, stick it onto this histidine, and then that phosphorylates the first component, the sensor kinase. The sensor kinase then transfers this phosphate to OMP-R, the response regulator. And OMP-R phosphate then can become a dual activator and repressor of the appropriate genes. In this particular case, OMP-R phosphate activates, so sits on the promoter of OMP-C to allow for more OMP-C to be expressed. Simultaneously, it sits on the operator of OMP-F to block OMP-F expression. So that in that way, your progeny can get more OMP-C produced as you grow and less OMP-F as you grow in this high osmolarity environment. Another type of gene expression regulation is quorum sensing. It's cell-to-cell communication mediated by small signaling molecules, such as N-acyl homoserine lactones. So N-acyl homoserine lactones are primarily used by gram-negative bacteria, but there's a bunch of different kinds of them, and there's different signaling molecules that microbes will use. Some are just short peptides. But quorum sensing, as a general rule, couples cell density to transcription regulation. And so here's a cell at low cell density, and it produces a few of these homoserine lactone molecules, or signaling molecules. We also call them pheromones. So imagine these red little triangles as pheromones or N-acyl homoserine lactones if they're gram-negative bacteria. But these little molecules, there's not very many of them, and they kind of diffuse off into the world and don't do much. But when there's a high cell density, so you see lots of cells, each of these cells producing some number of pheromones, the pheromones can go into and out of the cell, and they can transfer into their neighbors as well. And so that creates a group behavior complex. And so in this particular case, imagine luminescence is a possibility where the microbes can actually activate gene expression together and produce a luminescence response. So what you see here is a flask of a pretty high concentration of bacteria, and these are bioluminescent bacteria that are regulating the bioluminescence by quorum sensing. They're literally sensing their own cell density and then activating expression of bioluminescence genes. You can't see it right here because the culture is in the light, but once we turn the lights off, you can readily see the bioluminescence. And by swirling the flask, you're adding oxygen, which enhances the biochemical reaction. And so by that, they're doing that by quorum sensing. So there's a couple things going on. In that flask, because there's walls around the flask, these little pheromones can build up. So if this guy's just, a single bacterium swimming around in the ocean, these little pheromone signals are going to diffuse away. But if the microbes are in a flask, especially, they can build up and activate each of these cells to individually make light. And collectively, the collective light of billions of cells can actually be visualized. In nature, we mostly see this bioluminescence produced by bacteria when they're in a symbiotic association with certain animals, like flashlight fishes or certain species of bioluminescent squid, which is what I do some work on. And so here's just an overview of the genetics of this process. And so here's a DNA fragment showing you what's called the Lux operon. And so this operon consists of Lux I, Lux C, D, A, B, E, G. So it's a series of multiple genes that are necessary to make this light production. And so the microbes have positive regulation. In this particular case, they produce a transcription factor called LuxR in its inactive form. And as the bacteria are producing lots of these pheromones and others around them are making the pheromones go into and out of the cell. And some of those pheromones will find LuxR and bind to LuxR and make it active so that LuxR can bind to the promoter and allow RNA polymerase to transcribe all of these luminescence genes. And so in that way, the microbes are able to figure out how many siblings are around them in order to activate gene expression and perform some kind of group behavior. So I just talked about bioluminescence, but you can imagine that this process could be really important for virulence too. Imagine these are bacteria that are in patients' lungs, and you wouldn't want to produce a bunch of toxins if there aren't very many of you, because that could irritate the lung and cause an immune response. And so you might want to secretly and silently grow and produce pheromones. And then once you've got enough brothers and sisters around you, you can then rapidly start producing toxins that could then damage the system without causing an immune response when your numbers are too low.